Audio recording of lecture 9, double-stranded RNA viruses. The topic of this lecture is replication of double-stranded RNA viruses, and we also have a section that talks about subviral pathogens. Slide number 2, the first family of double-stranded RNA viruses are called family Reoviridae, Reo respiratory, enteric, orphan. So they cause respiratory infections, enteric or GI tract infections, and they are also considered as orphan viruses. That orphan viruses are those viruses that are not associated with any specific diseases, but they have they are pathogenic, so they possess pathogenicity, meaning that they have the capacity to cause damages to the cells, tissues, and organs. So this family, Reoviridae, the genome is double-stranded RNA, positive and negative sense, or plus and minus, or sense and antisense, and the genome is segmented, so multipartite. It has a double icosahedral capsid. We have an outer capsid that has very little short spikes on it, the blue portion that we see in this slide. And the inner core capsid contains nucleocapsid, those proteins that are associated with the nucleic acid or the genome. It has a very wide host range. Reoviridae family of viruses can infect plants, insects, and animals. So slide number 3, I have provided a little bit of an additional information on Reoviridae and the type of diseases that they cause. It's basically what we just discussed, but I would strongly recommend you to take a look at this slide to become more familiar with the details of this virus and how what's the pathogenicity of the virus, how it infects various tissues and organs, and what are the diseases caused by the virus. A genus within Reoviridae family is the genus Orthoreovirus. Orthoreovirus infects avian, mice, humans. In humans, it causes a mild upper respiratory tract and GI disease, both mild upper respiratory tract, the respiratory illness, and the GI disease, the enteric illness. And the route of transmission of orthoreovirus is fecal oral route of transmission. Moving to slide number 5. The next genus is called rotavirus, rota. So they have wheel-like spokes. They can also infect animals and humans. Fecal oral route is the route of transmission, mainly through respiratory secretions. And rotaviruses cause infantile diarrhea in infants, and gastroenteritis, usually in children under the age of 5 years. In the United States, we have winter epidemics of rotavirus, but now it's a lot less because of successful vaccination practices. And around the world, rotavirus can cause epidemics, and in developing countries, greater than 1 million infants will die due to rotavirus infection per year. Slide number 6, the next genus is called Colty virus, Colty for Colorado, tick fever virus. That's the main virus in this genus, Colorado tick fever virus. As the name implies, transmission of this virus is by tick bites to animals and humans. It usually causes fever, headache, and sometimes severe myalgia, which is muscle pain. I have a little bit of a description of myalgia down here at the bottom of this slide. And Colorado tick fever virus infection may also lead to meningitis and encephalitis. Let's take a closer look at human reovirus. As we mentioned before, it is has a double-stranded genome, double-stranded RNA, both positive and negative sense, and it's segmented, so it's multipartite. These segments, we have 3 L segments, L1, L2, L3, 3 M segments, M1, 2, 3, and 4 S segments, as we can see in the picture on the right-hand side. I would strongly recommend you, when you're looking at this slide, enlarge it a little bit so you can easily see different mRNAs being produced from these gene segments. The genome is relatively large. The total genome is 23.5 kilobase. S1 gene, these 4 gene segments, one of them is S1. It's at the bottom of the slide here. S1, 2, 3, 4. And S1 messenger RNA has 2 overlapping translational open reading frames, or 2 ORFs, that these have alternate initiation site codon, meaning that they have both, they're overlapping, they have 2 regions where translation can occur, therefore it results in the production of 2 different proteins. So S1 produces 2, and the other 9 segments will produce 1 each, a total of 12 different viral proteins, and they are named as lambda, mu, sigma, large, medium, and small proteins. Slide number 8 is just basically a depiction of what we just discussed, with a little bit more emphasis on the S1 segment, which is this segment due to having 2 overlapping open reading frames with different initiation start sites. It codes for 2 proteins. And slide number 9, for those of us who may have forgotten about different Greek alphabets and the symbols, I have included this slide so we can remember, as we just talked about in the previous slide. We talked about sigma, then we had mu, and we had lambda. So let's see, what are these things? The reovirus outer capsid protein, as shown on slide number 10, codes for 3 types of proteins, or it's made up of 3 types of proteins. These are sigma 1 dimer, mu 1C, and sigma 3. Sigma 1 dimer, or hemagglutinin, it participates in the attachment of the virus to host cell receptors, and it also inhibits host cell DNA synthesis. Mu1C activates viral RNA polymerase enzyme, the enzyme that is required for replication and making more of the viral double-stranded RNA genome. So the genome is RNA. We need an RNA polymerase to make more of the RNA. And the last one is sigma 3 that inhibits host cell RNA production and subsequent protein synthesis. So if you have these proteins that like the sigma 1 dimer that inhibits host cell DNA synthesis, and sigma 3 that inhibits host cell protein synthesis. Therefore, the host cell cannot survive. After RNA is being replicated, the host cell will die. Whereas real virus core proteins are usually those enzymes that are required for RNA synthesis, as depicted in slide number 11. These include lambda 1, sigma 2 complex, that is a polymerase, lambda 2, which is a capping enzyme for the 11 messenger RNAs is being produced, and lambda 3 is also polymerase. So core proteins are usually, those core proteins are usually a bunch of enzymes, polymerase, and capping enzymes. So let's see slide number 12. Let's see how is the reovirus entry and partial uncoding. Entry of reovirus is through receptor-mediated endocytosis. Upon entry, lysosome is being formed, then lysosomal fusion results in degradation of the outer capsid proteins of the virus. Once it's degraded, the infectious subviral inner core particles will be released from the lysosome into the host cell cytoplasm. Then we have in the host cell cytoplasm, we have double-stranded RNA, we have core enzymes, lambda 1, sigma 2 complex, lambda 2, lambda 3. These are enzymes required for capping and replication, polymerases. And then we also have mu13 that it basically causes the activation of RNA polymerase. So it all sets the stage for replication of double-stranded RNA genome of the reovirus. Slide number 13 talks about conservative mRNA transcription. Basically, what will happen is as depicted in the slide. We have reovirus attachment to specific host cell receptors, gets inside of the endosome through so-called receptor-mediated endocytosis, changes in the pH, influx of hydrogen ions, acidic pH results in uncoding, release of core enzymes in the host cell cytoplasm, and then some of them are capping enzymes. So we have messenger RNAs are being produced and they are being capped. So once the inner core particle of the virus is being released inside the host cell cytoplasm, the mRNA transcription will happen. It's a double-stranded genome, so it needs to unwind and open up, and this is accomplished by viral helicase. Double-stranded, the messenger RNA of the virus is being copied from the negative strand, and these daughter messenger RNA uh, that are being produced, they exit through vertices into the cytoplasm, and the double-stranded RNA remains inside of the core particle. Only the messenger RNA comes out from the core particle, but the double-stranded will remain there. Slide number 14 is just basically a depiction of what we just discussed. Slide number 15 is a three-dimensional visualization of where the mRNA in purple is being released from the rotavirus particles. So mRNA is being produced and then it will be released. Slide number 16, once the messenger RNA is being produced, it needs to be translated. So once the mRNA from the inner core particle of the virus gets released into the host cell cytoplasm, it will immediately be transcribed on host cells. It will be immediately translated on host cell ribosomes. And we have a very regulated gene expression. There are different sets of genes being expressed as a result of translation of different mRNAs. Initially, we have 4 early mRNAs, it's just the timing, that these mRNAs code for non-structural proteins. Remember, we have early gene expression, genome replication, and then late gene expression. So early gene expression is the step immediately prior to genome replication. So what we make at this stage are non-structural proteins, mainly enzymes required for genome replication. After non-structural proteins are being made, genome is being replicated, then we have 6 late mRNAs that code for non-structural as well as structural proteins. So genome replication, these late structural proteins that are being made as a result of translation of late messenger RNAs will assemble into developing inner core. So the inner core is being developed, so we will basically assemble these different proteins. So we have 10 viral mRNA gene segments that are being inserted into inner core, and what will happen is we start copying negative RNA strand on viral mRNA. So mRNA is usually a copy of the positive strand. We have negative strand in order to make double strand, that RNA genome of the virus. And the inner core that now has the double-stranded genome and has all of these viral proteins needed will be used for mRNA transcription. And it can also, because of the double-stranded genome, it can be incorporated into the progeny virions. Reovirus assembly and release. Outer capsid forms around the inner core. So we have the genome, then we have an inner core around it, and then an outer capsid around the inner core. So technically, we will end up with double capsid. And the release of reovirus from host cell is through cell lysis. Therefore, the host cell will die after the virus is being released. So let's talk a little bit about subviral pathogens. Subviral pathogens can be divided into defective virus, viroid, and prions. Defective virus is a virus that is unable to replicate on its own. Example would be hepatitis delta virus, HDV, that by itself cannot replicate. It requires a helper virus to replicate, hepatitis B, which we will discuss. Another group of subviral pathogens are viroids. These are very small RNAs. Example would be potato spindle tuber viroid or PSTV. And last group of subviral pathogens are prions. These are proteinous infectious particle, and they can cause diseases such as Kuro or Creutzfeldt-Jakob disease. Slide number 20, let's talk about defective virus. Example would be hepatitis delta virus. It has a negative RNA genome that is complex with hepatitis delta virus protein, or so-called delta antigen. Delta antigen. And this delta antigen has basically an envelope. The envelope comes from the hepatitis B virus. It is called hepatitis B surface antigen. So it's on the surface, S. And since it comes from hepatitis B, HB, and it's an antigen, meaning that it can be recognized by the immune system. So let me enlarge this picture a little bit so you guys can see that a little bit better. So yes, this is the hepatitis B surface antigen, hepatitis delta antigen, and it has a circular single-stranded negative RNA genome. HDV infects about 15 million individuals around the world. So if we have help along the infection, then about 40, which happens in 40% of these 15 million. So it ends up in fulminant hepatitis. On slide number 21, I have provided a little bit of an information about hepatitis, the way that we can prevent it or cure it, etc. And I would recommend you to take a look at it and become familiar with it. Slide number 22 talks about HDV genome, which is a single-stranded negative sense RNA genome. Again, it's a single-stranded, it's circular, it's rod. It become, even though it's circular, but due to base pairing, it looks like rods. It's just actually just a complementary base pairing, and it's relatively small, 1.7 kb. This genome is very similar to RNA of viroids. HDV is a defective virus. As we mentioned, it is incapable of self-replicating. It only replicates in the presence of hepatitis B virus infection or HBV infection, because HBV provides replicative functions and viral envelope for HDV. So it basically helps it out. So HDV RNA synthesis, what will happen? Once it gets into the cell and there's separation of nucleic acid from the capsid, the negative RNA genome that is released, as well as the associated delta antigen, will be transported into the host cell nucleus. It's negative, it makes positive anti-genome, we have genome, it makes positive anti-genome, and the production of positive RNA is mediated by host cell RNA polymerase 2. And then later on, the messenger RNA will be uh, it will be transcribed. Sorry, let me take a step back. We have negative genome, we make positive genome by host cell RNA polymerase 2, and the host cell RNA polymerase 2 can also transcribe host cell mRNA. So this is one type of RNA, a second type of RNA, and then we also make a subgenomic messenger RNA, which is smaller segments of the original RNA. And this subgenomic mRNA is made via two mechanisms. One mechanism is called interrupted anti-genome transcription. So different regions of the anti-genome will be transcribed, the positive RNA, or it can be uh, made by autocatalytic ribozyme activity of circular RNA. But basically, the RNA that we make on there, it has a self-cleavage site that through an autocatalytic activity mediated by an enzyme called ribozyme, it will come and this circular RNA will be converted at the bottom to a linear mRNA. So we have these three types of RNAs. The most abundant one would be the negative sense genome, like 300,000 copies per liver cell. Then via the action of cellular RNA polymerase or RNA pol 2, we make a positive copy of this negative strand, and we have about 50,000 of these positive sense antigenomes per liver cell. And both the negative sense and the positive sense genome have these self-cleavage sites that ribosome can cut it and make a linear mRNA. So these are the three types. On slide number 24, I have discussed subgenomic messenger RNAs. For those of you who may have forgotten about some of these concepts, exons, introns, transcription, template, ligation, etc., I would strongly recommend to look at it at this slide. Slide number 25 is just basically what we just talked about, how the three types of hepatitis, three RNA types of hepatitis delta virus are being made, that we see them in infected liver cells or hepatocytes. As we mentioned, one is the negative RNA, one is making a positive RNA with the use of host cell RNA polymerase 2, the number of which is a lot smaller, and through two different mechanisms. One is interrupted replication of the positive strand, or through self-cleavage. At the self-cleavage site, we will end up with a messenger RNA that it has one open reading frame that codes for small delta antigen. The number of this last messenger RNA is very low per liver cell, whereas the negative sense genome has the highest number and the positive sense has the middle range. Negative sense 300,000, positive sense 50,000, and the mRNA 600 copies only. There's another method that I did not discuss it, and that is the RNA editing part. There is a site called RNA editing site, and what will happen is RNA will be edited by host cell enzymes. So the translational termination sites will be altered. And if that happens, then we end up with two proteins. One is slightly longer. I believe it's 19, 20 amino acids larger and longer than the unedited form. So, but I want you to concentrate on how these three types of RNAs are being produced. Slide number 26 talks about hepatitis delta virus disease. HDV is transmitted by blood and body secretions, very similar to the transmission of hepatitis B virus and hepatitis C virus. With HDV, we have two types of infection. One is called co-infection, the other one is called superinfection. Co-infection is simultaneous infection of HDV and HBV. This results in acute HBV infection because HBV brings in the HBs antigen. Then the body can produce protective antibodies against HBs antigen. So it's anti-HBs antigen antibody. So basically, an antibody against this. Sorry, it makes an it makes anti-HBs antigen antibody. It makes an antibody that is against HBs antigen. So co-infection results in a less severe type of disease due to the fact that we have protective antibodies. Whereas super infection or UPN means that we have uh, uh, a chronic HBV, we'll see superinfection of chronic HBV patients. What will happen is patient has HBV, HDV will come in and will be added on top of it. Unfortunately, protective antibodies cannot be made. The disease is a lot more severe and it's persistent. It leads to fulminant liver infection and has high fatality rate. Because this superinfection is a chronic type of HBV infection that has happened over an extended period of time, it increases the risk of severe liver damage, a condition called cirrhosis, and also makes the patients a lot more susceptible for liver cancer or hepatocellular carcinoma. So the infection is a risk factor for hepatocellular carcinoma and cirrhosis. Slide number 27, let's take a look at the HDV life cycle. As we mentioned, virus comes in, there would be an uncoding of negative or single-stranded RNA genome from the capsid proteins. Then we have the synthesis of a positive RNA. Negative RNA is genome, positive would be anti-genome, as well as making messenger RNA. The double-stranded positive and negative will be used for as the progeny for those RNA virus, RNA as the progeny, as the negative RNA genome for the progeny viruses. How? The positive sense part of it will make messenger RNA, will make proteins, and including the delta antigen, and the negative sense is the nucleic acid. So the positive, the products of translation of these single mRNAs will associate with the negative genome to form ribonucleoproteins. So we have the genome, we have the proteins, the two will associate with each other to make ribonucleoprotein. Then the hepatitis B surface antigen that will be incorporated into the envelope or outer portion or region of the virus will be picked up from the host cell endoplasmic reticulum, and the cell will be secreted. The virion or viral particles will be secreted out of the host cell. Slide number 28 talks about another subviral pathogen called potato spindle tuber viroid or PSTV. This is an infectious single-stranded negative RNA. It looks circular due to self-complementary base pairing that looks like a rod structure, and it gives the impression that it's a double-stranded RNA. But in reality, it's a single-stranded that has basically base paired with itself through self-complementation. The genomes are 250 to 316 nucleotides. This RNA genome appears to encode no proteins and is capable of autonomous replication. So it can make more of itself. And genomes all contain 5 regions. 1 region, 2, 3, 4, 5 regions. These regions are called domains. So let's take a look at them. We have the left-hand side region, the right-hand side region. On both ends, we have left and right-hand terminal domains, terminal 1, terminal 2. Then we have a region called pathogenic domain, or P. In the center, we have a conserved central domain. Then to go closer to the right-hand side, we have a V or variable domain. Okay, so let's see what's going on. Remember, I mentioned that viroids appear to be capable of autonomous replication. So let's see how the viroid genome replication will happen. Once it infects a cell, it uses host cell RNA polymerase 2 and double-strand helical arrangement of viroid RNA competes effectively with host cell DNA for RNA polymerase 2. So that's basically what it's saying, is that RNA polymerase 2 participates in the replication of your genome. We may also receive some help from host cell RNA polymerase 1. And then once the RNA is being made through a process called autocatalytic or self-cleavage, it can be chopped off or cleaved into unit-length RNA infectious form. These are those RNAs that can infect host cells. The disease is caused by viroid. Viroids usually destroy important crops. They're transmitted from plant to plant through mechanical damage, insects, seeds, or cuttings. An example would be potato spindle tuber viroid that was discovered in 1971 by United States Agricultural Research Service. And these tubers of potato were narrow, as you can see, and long compared to the uninfected. We also have another type of viroid called chrysanthemum stunt viroid that affects these flowers. They have the infected plants have shortened stems, uneven flowering, and smaller foliage. Slide number 32 talks about the potato spindle tuber viroid. Please take a look at it. Slide number 33 talks about viroid pathogenesis. How does a viroid cause damage? The P domain or pathogenicity domain of viroid is complementary to host cell 7S RNA. This 7S RNA of the host cell is involved in protein translocation. So it's being postulated that this viroid that hybridizes or associates with 7S RNA because they're complementary to each other, this will disturb proper transport of host cell proteins. Once the proper transportation is being disrupted, this results in alteration in plasma membrane structure that we see in viroid infection. Remember, on the previous slide, I mentioned that the tubers of potato are narrow and long due to disturbed and disrupted protein translocation. The last group of subviral pathogens that we'll talk about are prions. They do not have any nucleic acid. Therefore, the ability of these particles to cause infections or their infectivity is not inactivated by those enzymes, such as DNAs or RNAs that cleave off or chop or deactivate DNA and RNA respectively. So it's not nucleic acid, therefore these enzymes won't work on it. We have an infectious protein that this infection protein can be destroyed by proteases under normal circumstances. These proteins have long incubation period in host. Usually they can stay up to 30 years. And in the past, they used to be called unconventional slow viruses due to their long-term incubation period. So how do we test it? We treat the tissues, the cells, with an enzyme called proteinase K. The test is called proteinase K digestion, and we use a technique called Western blot analysis. So what will happen? If you have a host cell protein, this host cell protein, normal protein, is sensitive to this proteinase. It's an enzyme that chops off or digests or cleaves protein. Normal proteins are sensitive to it. So if we treat the cells, then run a Western blot, we don't see anything but because the protein is being destroyed. However, those prion proteins that we see in various diseases, CJD, we'll talk about it, Creutzfeldt-Jakob disease. This protein, which used to be a normal cellular protein but has misfolded, is resistant to degradation by proteinase K. Therefore, we see a band. It won't disappear on a Western blot. So again, if we have different tissues that we're suspicious of whether they're normal or diseased tissues from usually brain, then we treat them with proteinase K, and the proteinase K, if it's normal brain, we don't see anything on the Western blot because the protein, normal host cell protein, is sensitive to this enzyme, whereas the infectious proteins are resistant, so it won't be affected. So let's talk about a little bit about this prion protein. We have two types of proteins. One type is the normal brain protein that is a small protein, 27 to 30 kilodalton. But then we have a protein that this protein is resistant, RES, resistant to the enzyme proteinase K. So proteinase K resistant protein, this is basically an isoform of the normal brain protein. However, it has unusual folding pattern. And it's because of this unusual folding pattern that is infectious, and this is the pathogenic form of the protein. Using electron microscopy, if we look for those proteins that are resistant to the enzyme derived from brain of patients, we see those proteins as macromolecular fibers. They look like fibers, and what will happen? They will, since they're in the brain, they interfere with the normal function of the neurons or brain cells or nerve cells. We have different types of prion diseases. These are in mammals, we see spongiform encephalopathies. In sheep, they cause a disease called scrapie. In cattle, we have BSE, bovine spongiform encephalopathy, or mad cow disease. In humans, we have a disease called Kuro. Kuro is a shivering. The patients shiver all the time. It is seen in New Guinea, and another disease called Creutzfeldt-Jakob disease, or CJD. Slide number 37 is a little bit of a description about these different types of diseases that we see: Creutzfeldt-Jakob, Kuro, Scrapie, and BSE, or bovine spongiform encephalopathy, or mad cow disease. Slide number 38, Alzheimer's disease, link to prion. Alzheimer's disease, as we know, is a severe disease that is characterized by loss of mental function and loss of memory. It decreases thinking process, and it is also associated with some personality changes, mainly due to disruption of mental function and thinking process. In the in the brain of those patients with Alzheimer's disease, we see something called amyloid plaques. These are basically neurofibrillary tangles due to proteins called tau proteins. That this process of amyloid plaques is very similar to what we see in prion diseases. Therefore, scientists think and are suspicious that brain diseases such as Parkinson's and Huntington disease, because of this link between Alzheimer's disease and prion, they think that other brain diseases such as Parkinson's and Huntington's disease may also be due to an infection by a prion. Summary: reovirus is unique in having a double capsid structure, inner core, outer core, and it has a double-stranded RNA as its genome. RNA transcription occurs in an inner core particle formed by breaking up of the outer capsid, and then after the messenger RNAs are being made, they leave the inner core, they go into host cell cytoplasm. There are some pathogenic agents that are smaller than a virus, and they're very different than a virus. These include defective virus, incapable of replicating by itself, viroid, infectious RNA molecules, and prions, or infectious protein molecules. And I'm not going to go over the possible origins of virus, etc. This would be the end of lecture on double-stranded RNA virus, lecture 9, and subvirals, actually. Thank you very much for your attention.